So we know that all the genetic information, it is present in DNA, and this DNA will make the RNA cistrons, and that will produce the particular protein. So if it is a normal DNA, it will make normal mRNA, and that will produce a normal good protein. But if something, a defect happen in this DNA, that could lead to change in the sequence of the nucleotides, that will give result in change in the mRNA, and finally we'll be getting the abnormal protein. Okay. Or you will not at all get any protein. So there is a normal gene will give the normal protein. If it is a mutated gene, it will give the abnormal protein or no protein at all. Okay. So mutated gene either codes for the abnormal protein or it doesn't code for any protein. Okay. So we can say from this, what is mutation? Mutation, it is a phenomena where that will result in alteration in the DNA nucleotide sequence that will change the either the genotype or phenotype. So what is mutation? The change in the genetic code which result in change in the proteins. So that will induce the effect in the genotype or phenotype. So mutations occur either at the genetic DNA level or it could even happen at the chromosomal level. If it is DNA level, there is a few nucleotides will change here. So either a nucleotide is added or either the nucleotide is removed, or in place of other nucleotide, some other nucleotide will be inserted, substituted. Okay. Whereas in the chromosomal level, either the patch of the chromosome can be deleted or it is altered. So here it will produce a larger effect in the chromosomal level, whereas the DNA level, it will induce few changes or alterations in the nucleotide sequence. So based on that, we can classify the mutations into two types. One is that happening in the DNA level. The other one is happening at the chromosomal level. So it's called as the chromosomal mutations. And something that's happening at the DNA level, it's a genetic mutations. So chromosomal mutations. So we know in addition to recombination, what happens in the recombination? There is a male genes and the female genes. So that will be inherited to the offspring. So you will be seeing the change in the gene or change in the nucleotides that will result in different phenotype. That will be helpful for evolving the organism. But other than this change happened due to the recombination, the change can also happen from the mutation. Okay. So one is through the recombination, we will be seeing the variation in the genetic material. Apart from the recombination, we can also see the variation happen through the mutations. So mutations is the another phenomena that could lead to variations in the DNA. Okay. So the DNA, we know it is a helix and it was tightly so super coilly packed in the form of chromatids. And these chromatids could be heterochromatin, euchromatin, and finally they will be arranged into the chromosomes, which is a very compact arrangement. So that's happened through the condensation of the genetic material here. So when this happening, when this packaging happening, there could be the genes or loss. So deletion of the genes could happen, or some extra genetic information has added up. So there is a gain by insertion or duplication of the genetic material of the segment of the DNA. So that gives result in the alterations or changes in chromosomes. So that mutation is called as the chromosomal mutation. So what's happening in the chromosomal mutation? So there is a loss, that's a deletion of the genetic information, or extra genetic information is added in the few segments of the DNA, which will induce the changes in the chromosomes. It's called as the chromosomal mutations. Okay. So since the genes are known to be located on the chromosomes, so any alteration in the chromosome result in causing the abnormality or aberrations. Aberrations, in the sense, it's the synonymous to abnormality. The changes happened in the genetic material. So they refer to the change in the structure of the chromosomes or the number of the chromosomes. So this genetic disorder, we call it as the aberrations. So remember this. Okay. So it could leads to the change in the phenotype of the organism, change in the genotype of the organisms. So chromosomal aberrations are commonly observed in cancer cells. So whatever the mutations that induce cancer, it's mostly due to the chromosomal aberrations. And the mutation happened at the gene level, that's the DNA level. We have seen sickle cell anemia in molecular basis of inheritance. So the mutation arises due to change in a single base pair of the DNA. So if it is happening for one nucleotide change, then that mutation is called as the point mutation. You can take example that we have studied in the molecular base of inheritance, sickle cell anemia. So what happens here? Either that single nucleotide is deleted, or in place of that single nucleotide, another nucleotide could be inserted, or some other nucleotide is added as extra. There could be three possibilities. So all these three possibilities could cause the frame shift mutations. That means whatever the codons, we say the three codons, either they move forward or backward, and that could influence the overall change of all the codons. So that's called as the frame shift mutation that we have studied in molecular base of inheritance. So there are many chemical and physical factors that could cause these mutations. So all these chemical, physical, biological factors that are causing the mutations, we call them as mutagens. One of the example here is UV radiation that's causing mutations in the organisms. Mostly that will cause the skin cancers. So the mutagens can be physical agents, biological agents, chemical agents. Here, for the physical agents, UV rays, biological agents, it could be virus, bacteria, and chemical agents, it could be the cigarette, benzoyl peroxide, all the chemicals. So, what is the answer for this? Change in the sequence of nucleotides in DNA is called. What is the answer for this? Change in the sequence of nucleotides in DNA is called.
Mutation.
Yeah. So it is the mutation. So pedigree analysis. Pedigree analysis, it's nothing but the study of particular trait that is inherited from one generation to the other generations. So we call it as the pedigree analysis. That means how the mutations were inheriting from the parents to the offsprings, from one generation to other generation. So that studying, we call it as pedigree analysis. So it will give understanding how the pattern of this inheritance is happening, whether the mother is a carrier or the father is a carrier, whether they are uh, seeing the mutations in them, whether they have a physical change or not. So all these analysis will be given by this pedigree analysis. Okay. So it also helps to know whether the trait is dominant or recessive as well. So here the mutations also depends on same like the principles of inheritance. So the mutations are also carried the same way. So here we can see the dominant trait will be affected with the mutations. The recessive trait cannot be affected with the mutations. So such scenarios we can see. So in order to study such scenarios, this pedigree analysis is very helpful. So in human genetics, pedigree study provides a very strong tool to trace the inheritance of a specific trait, abnormality, or disease. So you can see all these things. With the help of pedigree analysis, we could able to study the specific traits, the abnormalities, or the disease patterns that is inherited from the ancestor to the offsprings, from one generation to the next successive generations. So here in the pedigree chart, we'll be using certain standard symbols in order to study. So this is the male. If the square box will refer for the male and the circle will give the female. And if it is we do not specify the sex or we don't know which sex it is, then we'll be giving like a rhombus, which is a sex unspecified. And if the species got affected with the disease, then we'll be marking with the red color. So the coloring will refer for the affected individuals. And if it is drawn a line between the square and the circle, that means they both are mating. So they are the couples, they both are mating. And if it is double, mating between the relatives. So that's a consanguineous mating. That means both are closely genetically related. And it can also show the parents. So it can also show the parents and the family. So the line drawn between the two, it will refer for the parents, and below it will refer for the children. So it could be the girl childs, or it could be the boy childs. Okay, it could be the male as females. So we'll refer with the circle or the square. So parents with a male child affected with the disease, then we'll be referring. If it is a female child, then we'll be showing with the circle and drawn inside, shaded inside. And if it is like this, it is a five children, but we don't know whether they are male or female. It's just a unspecified year. And it's unaffected because I didn't share anything here, shaded here. If it is shaded inside, then we'll represent it as a affected. So pedigree,
So,
yes.
so can you explain the pedigree analysis like this briefly again? Sorry, like I actually had some network issue.
So these are the symbols. You mean this one or complete pedigree analysis, what it is?
No, so the next slide, the
Okay, here
illustration.
we are using certain symbols to refer the characters. First, suppose if it is a male, then we'll represent with a square. If it is a female, we represent with a circle. If it is gender is not specified, then we'll refer with the rhombus. And if it is the individual got affected, then will be shaded inside. So you can see here, it was shaded and this was not shaded. So affected individuals will be shaded inside. So this one will be the affected individual. So genetically affected, the one which is having the mutation seen. And if you are showing the couples which are having the mating, then we'll be drawing the line between the two. And if both are related to the same, genetically closely related, then we'll be drawing the double line. And we can also represent the parents and children within the same by showing on the uppers of on the above we'll be showing the parents, below we'll be showing the children. If it is a male, we'll be showing with the square. If it is a female, we'll be showing with the circle. If we are shaded this one, then we'll tell that it is the affected female. If I shade for the male, then we'll say the children. Here, the male child was affected with the mutations. So you can see here, the parents with a male child affected with the disease. And if we don't know how many offsprings are there, sorry, we can also represent by the number inside this box, which will tell about those many number of offsprings are there. And if it is all are male, then I'll represent with the square. If it is all are female, I'll represent with the circle. If I don't know whether it's a child, the child is it's a male or female, then I'll represent with the this one. So is that clear?
Yes, sir.
Okay. So what is the pedigree chart will do? It will represent a family line from one generation to the next generation. So it will give a family tree. So in that family tree, who got affected and who doesn't, and not I mean not affected with the mutations. So if it is affected, then we'll be shading the box. If not, we'll not be shading. Okay. So that will give a pattern of flow of these mutations from one generation to other generations. So family chart showing the transmission of a particular trait or disease. For suppose here, there are first generations which have three kids, I mean three pairs. So in that, which one you can see here, this is a female, this is a female, and this is a male. The male got affected. So this first generation couple are having three kids. One is a female, the other is female, the other one is male. So here the male is affected. So I represented with the orange or colored one. Okay. This male got married and their children doesn't show any mutations. And here the couples doesn't show any mutations. Here their children show mutations. That means here the female acts like a carrier. That means she is not affected with the disease, but she masked the disease in her genes. So it was shown in the next generations. So the female doesn't affect with any mutations, but her next generation got affected because she acts like a carrier, but she is not affected with the mutations. So we called her as the carrier mother. The same happened here. Okay. Here the daughter is also not affected, but after two generations it was affected. So they are acting like a carriers. Here the female and this female acts like a carriers. So we'll be studying in detail about when we talk about each of the diseases that are given in the NCRT. So that's how you will be analyzing here. This is the family tree, how we'll do the pedigree chart and how it will be very helpful for analyzing the mutations inheritance. So genetic disorders could be classified into two types. One is the Mendel disorders, the other one is chromosomal disorders. Mendel disorders could be the sex chromosomes or autosomes. That's all the vegetative chromosomes. Other than the sex chromosomes, whatever the chromosomes that are left over, they are called as the autosomes. So if the mutations happened in these autosomal level, or if the mutations happened in the sex chromosomes, like in the gametes. So Mendelian disorders are mainly determined by alterations or mutations in the single gene. So remember, this is very important. So most of the Mendel disorders are the changes happened in the single gene. It's not a multiple gene. It is not a polygeny. It's a monogeny. It's a single gene. So these disorders are transmitted to the offspring on the same lines as we studied, like the principles of inheritance. First, suppose capital T, small t, how it was given into the capital T, small t, and how it was carried to the next generation. The same way, it was also following those rules. The same line, how the principles of inheritance happened, the same way these also. First, suppose in terms of capital T, I can see the parent with the mutated, I can say capital M, capital M. The one which is not mutated, small m, small m. So like that we can give. That will be talking when we'll study about some of the diseases. And most common and prevalent Mendelian disorders that mentioned in the NCRT, it's hemophilia, cystic fibrosis, sickle cell anemia, color blindness, phenylketonuria, thalassemia. So these are the diseases that mentioned in the NCRT. And we'll study each of these diseases, how the mutations are there, and what the symptoms of these diseases. So Mendelian disorders can also be dominant or recessive. They also show this. So by the pedigree analysis, one could easily identify whether it's a dominant or recessive in this order. So whether it's a sex specific or not sex specific, that's autosomal chromosomes. So that's it. What about these diseases are there that we'll be studying in the next class. So once these diseases are completed, then there is a recombinations. Those are there. So mostly in two chapters, we'll finish off. In two classes, we'll finish off this chapter. Okay, guys, thank you. Is there anyone have doubts?